Hello, everyone, and welcome to Lecture 2 of Metabolism. We're going to be looking today at carbohydrate metabolism, and we're going to start with a stage 2 pathway, mostly because it's universal and ubiquitous. Every single cell has this pathway, and it's also the most well studied and best understood of all. Hopefully, we're going to be able to illustrate many of the principles that we reviewed last time and also get the sense of the definitions that we put forth in the first lecture. So again, keep in mind that first lecture we're going to be coming back to it again and again, putting another layer of meaning to each definition and principle and understanding the concepts that we reviewed in that lecture. To start today, as in all the lessons, here are my learning objectives for today. And my suggestion is that when you're done studying a particular lecture, make sure that you are able to actually meet all these objectives. And if not, do bring your questions to the discussion board or to our live Zoom meetings on Fridays and whenever we're going to have them on Mondays as well. So with these goals in mind, first a couple of definitions to review, and so that we're all on the same page. Carbohydrates are major sources of metabolic energy in humans, and they are all organic molecules with the general formula or of a hydrated carbon. So it's really going to be carbon and water, and it's going to be a very long molecule if it's a complex carbohydrate with this general formula. They're readily available as direct products of plant photosynthesis. And this is why I like to say that we live and die by the grace of plants. If it weren't for plants to capture the energy of the sun into carbohydrates, which we are consuming for our own metabolic energy, we wouldn't be here. So carbohydrates are a major food group in humans. The FDA suggests that we consume anywhere between 45 and 65% of the daily caloric intake in the form of carbohydrates. And they're also one of the most abundant form of metabolic energy. Grain production last year was in the gigaton mass. So humans are large consumers of carbohydrates and also large producers of carbohydrates by really agricultural means, by growing plants. They're also highly amenable for long-term storage and preservation, both in your fridge or in the pantry, but also within the organism. There are ways carbohydrates can be stored locally, as for example, glycogen in liver or in muscle, but they can also be made into lipids, which then have their own organ where they can be stored for long-term later usage. So there are a number of advantages to carbohydrates, of why they evolved to be really our main source of energy. Just a quick review, several representative carbohydrates from plant sources, that would be the starches, the majority of them in grains. They're a mix of polysaccharide composed of amylose, which is a linear polyglucose, and amylopectin, which is a branched polyglucose. And also, as a plant source of carbs, is sucrose, or you know it as table sugar in a lot of sugary drinks. It's a glucose fructose disaccharides, and hopefully you get that from fruits, veggies, and legumes, which is the healthy type of sucrose. We can also get carbohydrates in from animal sources, and that would be glycogen, which is a branched polyglucose, mostly from muscle and liver of mammals or other animals, and also lactose, which is a galactose glucose disaccharide, which we can get from milk and dairy. And the main product of major or complex carbohydrates breakdown is the the ubiquitous monosaccharide glucose, which is derived from the poly and disaccharides that we just described, and makes a large portion of dietary intakes of human. Importantly to remember, the freely circulating blood glucose is maintained at 5 millimolar in the fasted state. And I would say that perhaps one of the major goals of this section of biochemistry is that at the end of the metabolism, you can explain the various ways in which human organisms maintain their 5 millimolar glucose levels in blood in a variety of states. So for example, right after a meal or after prolonged absence of nutrient intake in the fasted state, or even in the starved state. So glucose in blood, 5 millimolar, maintained by a variety of tightly orchestrated mechanisms, and it's going to be our job in this course to really understand and be able to explain how that happens. Importantly, glucose is a metabolic source of energy for all cells. All cells on this planet have the metabolic machinery to metabolize glucose. So therefore, all of them use glycolysis to make energy from glucose. As an overview of carbohydrate metabolism, as we will be discussing it in this class, we will start, as I already mentioned, with stage 2 of carbohydrate metabolism, which is glycolysis, and it's going to refer to the metabolic degradation of glucose into pyruvate, with the purpose of glycolysis being making the ATP. And we're going to also talk about lactic fermentation. In lectures 3 and 4, we're going to talk about stage 3 pathways, and these I'm talking about catabolic pathways of carbohydrate metabolism. So stage 2 and 3 are going to really be the material that we're going to assess in the first exam of metabolism. And we're going to move then to stage 2 of catabolic carbohydrate pathways, which is digestion and mobilization from glycogen, and then to anabolic pathways of carbohydrate metabolism. And when I say anabolic, I'm referring to making glucose from non-carbohydrate sources, like for example lactate or alanine, and that is happening very specifically in liver and kidney. And we're going to contrast that with glycolysis that occurs in every single cell that has been so far studied or observed. And we're going to also talk about glycogen biosynthesis. So we are not going to talk about the stage 3 reactions, the metabolic pathways that make stage 3 of carbohydrate metabolism, and those would be photosynthesis, and they only occur in photosynthetic organism, plants, and they require chloroplasts, and it's a bit highly specialized pathway that we are not gonna discuss in this class. Because I'm gonna remind you, in this class we're gonna discuss central pathways of intermediary metabolism, and those are the ones in which primarily mammals, because we are in a medical school and we're concerned with those pathways that help health and prevent disease. So we are gonna discuss about anabolic pathways of carbohydrate metabolism only for stage 1 and 2, and that together with stage 1 of catabolic is going to be discussed in exam 2. So basically 2/3 of this entire class we're going to discuss carbohydrate metabolism, and hopefully by then you're going to put all kinds of reactions and enzymes and metabolites, and these are terms that we discussed in lecture 1 on all these pathways. For today, however, we're just going to focus on stage 2, on glycolysis and lactic fermentation. And without further ado, the next 3 or 4 slides, I will say, are fundamental to this course. I'm going to very much suggest that you make sure that you understand everything that is on them, because we're going to refer to them again and again, and we're going to make use of these concepts and information throughout this course. So for each pathway, I'm going to offer to you the definition, and in the case of glycolysis, it's a linear sequence of 10 consecutive reactions that accomplishes the oxidative degradation of monosaccharides with direct production of ATP. And I'm going to stop a little bit on this definition to highlight a few principles and issues that we discussed last time. It's a linear sequence, so it has a certain architecture, and its architecture is linear. It's a sequence. All metabolic pathways are a sequence, such that the product of one reaction is going to be substrate for next. So there's going to be a sequence of how many? 10 reactions catalyzed by 10 enzymes that accomplishes, so it's a purpose, it has a purpose, and the purpose of glycolytic pathway is the production of ATP from glucose. So basically, glucose to ATP and pyruvate, it's the chemical definition of glycolytic pathway, and the definition also includes the strategy for this pathway. We mentioned that all pathways have a single direction, that have some architecture, they have a strategy, and in the glycolytic pathway, the strategy is oxidative degradation. The substrate is mainly glucose, but we're going to see in subsequent lectures that fructose and lactose can also be metabolized or catabolized through the glycolytic pathway. And its purpose is to produce ATP directly from glucose, meaning in the absence of oxygen or in the absence of mitochondria. And in the several points here, I'm offering to you perhaps the most important things that we have to remember or understand and remember about glycolysis. Ubiquitous means that it's everywhere. Every single cell has the glycolytic pathway. Universal means that every single cell using the same chemical reactions and conserved enzymes. So the same reactions and conserved enzymes in all cells accomplish the oxidative degradation of glucose with direct production of ATP and pyruvate. And it doesn't matter that it's an yeast cell or a bacteria or a liver cell. Every single cell can do that, can do the oxidative degradation of glucose with the glycolytic pathway in the same standardized 10 reactions, 10 enzymes way. We already referred to it as a stage 2, and you will remember that we defined a stage 2 in catabolism as reactions or pathways that transform monomers to metabolic intermediates. And in this case, we're talking hexose, which is the monomer derived from carbohydrates, right? Complex carbohydrates. It's exclusively cytosolic, meaning all enzymes are in the cytosol. Its most important purpose is to produce direct, fast ATP at the expense of reducing NAD to NADH. It's generally catabolic, and in the previous slide, we saw it categorized with the catabolic reactions of the glycolytic pathways on the left-hand side. However, keep in mind, and we're going to talk about this in a lecture later this semester, they can have anabolic purposes, meaning its intermediates may be used in a biosynthetic way. So for example, a cell can decide, for whatever reasons it needs pentose for RNA biosynthesis, it can make it from glucose. So although glucose is degraded from C6 to C5, the pentose, the purpose of this oxidation will be to make pentose for RNA biosynthesis. So one can say that glucose is being used in an anabolic reaction. So one other principle that we talked about last time was about the fact that all pathways are very highly interconnected, coordinated, and integrated. And that happens because certain metabolites are part of several pathways, meaning they serve to intersect these pathways. And we call these nodes. For example, glycolytic intermediates can be also participate in lipid, amino acid, DNA, as we just said, pentose. So pentose can participate in RNA metabolism. Pyruvate can participate in amino acid synthesis. Pyruvate can participate in biosynthesis of lipids. So pyruvate is a central metabolic node because it participates, we're going to see, in a number of reactions. In other words, it can be substrate and or product to a number of enzymes, several enzymes, each concerned with a different pathway. Is glycolytic pathway, as we said in the definition, it's oxidative. What does that mean? It extracts electrons from the carbon bond of the 6 carbons, so there are 5 bonds, onto NADH, and the NAD that was used to extract these electrons from glucose must be regenerated if this reaction is going to be continuous. And remember, NADH concentration within the cells like to stay constant. In a way, you can think of NAD and NADH as really coenzymes that stay constant in the cell and therefore must be regenerated. And how is that done? Well, it can be done in 2 ways. Anaerobically, as we're going to be discussing today, locally in the cytosol by fermentations. And fermentations will require additional enzymes on top of the 10 that are in the glycolytic pathway glucose to pyruvate. So meaning from pyruvate, we're going to have to add fermentative enzymes, LDH, ADH, to regenerate the NAD and finish the entire pathway. Another way to regenerate NAD, it can be done aerobically. That is much more efficiently. However, it's also more sophisticated. It requires mitochondria, the mitochondrial respiratory chain enzymes, requires molecular oxygen. So that is done in cells that actually have this equipment. Best characterized metabolic pathway. More than 100 years of research. We know the 3D structures for all the enzymes. We know the chemical and enzymatic mechanism for all the enzymes. You have actually looked in the part 2 of this lecture with Dr. Kosman at several of these, and we're going to use as an example many more. This pathway and the study of this pathway has defined many basic concepts and principles for biochemistry in general, like, for example, and we're going to discuss this a little later in this lecture, the concept of isozyme. They have been discovered in studying the glycolytic pathway. The principle that an enzymatic activity can be encoded by multiple genes such that their structure, although similar, is distinct and can be regulated. And the last thing that I would say as essential to know about the glycolytic pathway, and we're going to spend quite a bit of time in discussing this, it's regulated. Like all metabolic pathway, the glycolytic pathway is also regulated. It is a very complex regulation, as you would expect for any energy producing pathway, and it's incompletely characterized, meaning there is still a lot of work to be done there, and perhaps that is something that you will choose in your career to do. So better understand the glycolytic pathway, especially when it comes to metabolic states that are associated with disease. For example, in cancer, glycolytic pathway research is very productive and useful. So with those definitions and principles, it will be very important, being in a biochemistry class, to not only refer to pathways as simply with words, but also with reactions where the natural law of mass conservation and spontaneity are preserved. So again, these are going to be concepts that we're going to emphasize again and again. But to begin, what are the linear sequences? What is the linear sequence of 10 enzyme catalyzed reactions that starts with glucose and ends with 2 pyruvates, 2 ATP, and 2 NADH molecules? And this is actually the simplified general equation or sum of glycolysis. However, if we are to account for carbons, for phosphates, for protons, for water, this is the more detailed and more accurate definition. Here, when we're saying we're starting with glucose and we obtain, after the metabolic pathway of glycolysis, we obtain 2 pyruvate molecules, 2 ATP and 2 NADH. It really focuses you on the principal metabolite. This is going to be a C that still incorporates carbons. It's going to be a C3 carbon. ATP, which is the purpose, the why this actual enzymatic pathway has happened. And to keep in mind that reducing equivalents have been extracted. So this is a more conceptual way of representing a pathway. This is the chemical way that takes into account conservation of mass and conservation, and we're going to also have to account for conservation of energy. We're going to discuss that in a second. So for now, just in terms of the chemistry of what's happening, substrate for glycolysis, we already discussed. It's mostly glucose obtained from the digestion of polysaccharides or from digestion of personal stored glycogen that we have accumulated from a previous meal. And we're going to discuss how that is being accumulated when we're talking anabolic reactions. However, other dietary carbs such as fructose and galactose can use the same pathway, and we're going to discuss how that happens. And also metabolites from lipid catabolism, like for example glycerol, or amino acid catabolism, like for example glycine, alanine, aspartate, glutamate, can enter the glycolytic pathway, not necessarily from the beginning, from glucose, but through its intermediates. So they're going to interface or node or interconnect at one of these 1 through 10 enzymatic reactions. Okay, so these would be substrates for glycolysis. In other words, glycerol, glycine, and these other amino acids can be used for energy production through the glycolytic pathway, can insert themselves into this pathway. Products of glycolysis, certainly ATP, that's the prize. It's used metabolically for anything that a cell needs or wants to do, including active transport, signaling, and we had a whole list in the previous lectures of means by which ATP can be used and is necessary basically for life of any cells and for life of the organism. What happens to pyruvate? Well, it can be used to regenerate NAD by anaerobic glycolysis and fermentations, as we're going to discuss today. Or pyruvate and NADH can be metabolized further aerobically through the subsequent pathways of pyruvate, dehydrogenase complex, the tricarboxylic acid cycle, and mitochondrial respiratory chain, and we're going to discuss that in the next 3 lectures. Remember, this option of aerobic metabolism requires intact mitochondria and molecular oxygen, and we're going to see exactly why that is. I mentioned already, and I'm going to probably say this many times, ratios of ATP and ADP, which can be quantified, as we discussed in the first lecture, as the adenylate charge of the cell, and the NAD and ADH ratio are kept constant. That are important markers of the homeostasis that a particular cell maintains. And deviations from optimal values represent powerful regulators on glycolytic reactions and in many other pathways. You can already imagine that if the ATP per ADP ratio is low, that's going to signal to all ATP using enzymes, and it would enhance the activity of pathways, or that will enhance flux through pathways that produce ATP. And the other way around, if the ratio of ATP per ADP is high, then flux through catabolic energy producing pathways is going to slow down. And again, we're going to use these words so many times that hopefully by the time we're done with metabolism, you will be able to explain this in your community and to your peers and be a science communicator. All right. So we talked about chemical strategy, and we also, it's an important purpose of this class to demonstrate at every opportunity that I have how life happens according to natural laws. We are special, but we're not that special. Everything that keeps us alive, everything that permits, sustain life has to abide by natural laws. And in metabolism, there are two natural laws that I'm going to refer to again and again. One is mass conservation, meaning what goes in has to come out. And the other one is the spontaneity, the directionality in which various reactions are spontaneous. Now, the combustion of glucose into pyruvate and ATP, or the oxidative degradation of glucose into pyruvic and ATP, is a highly favorable reaction. So if you just look at the free energy of glucose to the free energy of pyruvate, it's clearly a lot of energy is going to be produced. Some of it is going to be incorporated in the ATP, and some of it is going to be released as thermal energy, which cannot be used to do metabolic work anymore. And if you look at the entire pathway, you're going to see that although the entire pathway is energetically favorable, and sometimes we refer to that spontaneous, not all reactions are, because if we are to graph, for example, the internal energy of intermediates, here's glucose, glucose 6-phosphate, which is the second metabolite in the pathway, has more energy than glucose. So that is not going to be a favorable reaction. Further, fructose 1,6-bisphosphate has even more internal energy than glucose. So the first several reactions are uphill energetically, meaning glucose is being energized, is being made ready for combustion, if you want, and I'm using metaphors here. So the first phase is an investment phase where ADP is being consumed, it's being invested to phosphorylate first glucose to glucose phosphate, and then fructose to fructose 1,6-bisphosphate. Why would the cell do that? Why would the cell that needs energy already, right? This is why the glycolytic pathways has to go on to make ATP. Why would it be using ATP? Okay. And turns out that this is actually a general strategy and principle where the first several steps, at least one step in the very beginning of a pathway, is going to be energy demanding. Because it means it's going to be, it's not going to want to go, right? It's going to be a reaction that is going to be difficult to happen, meaning it's going to need energy from the outside, meaning it can be regulated. So this is an important principle where the majority of metabolic pathways are initiated by an input of energy at the step that can be regulated. And that is because such a step, then it can be turned on and off based on the cellular needs of the metabolic needs of the cell. Once that investment has happened, then reactions occur that will actually meet the purpose of the particular pathway. And in this case, the purpose of this pathway is to make ATP. And overall, it's going to be a good investment of 2 ATP to get 4, meaning overall, the glycolysis per glucose molecule will produce 2 ATPs. So in the second phase, it's the payout phase, and the energy in high energy phosphase will be used to actually drive ATP phosphorylation. And I'm using here the word substrate level phosphorylation, or the concept, which is a very important concept in metabolism biochemistry. Just to remind you, substrate level phosphorylation denotes a reaction where ADP is phosphorylated to ATP at the expense of hydrolyzing a high energy metabolite. In glycolysis, this is going to be 1,3-bisphosphoglyceride, or PEP. They're both high energy phosphates. And what is meant by high energy? We discussed last time, anything that upon hydrolysis releases more than the energy needed to phosphorylate ADP, which is about 30 kJ per mole. Succinyl-CoA is also a high energy compound that can be hydrolyzed to produce ATP. However, note that that is not necessarily a phosphate, right? It's a succinyl-CoA. The high energy bond here is an S-CoA, right? It's not a phosphate. Importantly, there are only 3 substrate level phosphorylation reactions that can basically form ATP based on some other high energy molecule. And the examples are going to be 2 in glycolysis, by the reactions catalyzed by phosphoglyceride kinase and pyruvate kinase, and the reaction catalyzed by succinyl-CoA synthetase in the TCA cycle. Importantly, these reactions can be replicated in a test tube. They do not require cellular structure. That is in contrast to the other 2 ways of making ATP. Just these 3 ways: by substrate level phosphorylation, oxidative phosphorylation, which happens in mitochondria, and photosynthesis that requires chloroplasts. These are the only 3 ways by which nature makes ATP: substrate level phosphorylation, oxidative phosphorylation in mitochondria, and photosynthesis in the chloroplasts. And we're going to see all the levels of all the examples of substrate level phosphorylation, and also we're going to contrast that with the intricate, complex, and sophisticated process by which ATP is made in the mitochondria. So with further ado, these are the 10 reactions and 10 enzymes of glycolysis. And I'm asked every year whether these have to be memorized, and yes, you have to memorize all the metabolites, all the reactions, all the enzymes, be able to do a mass conservation and a energy conservation for the entire pathway, be able to explain how why the entire pathway is spontaneous, even if some of the reactions are not necessarily favorable. So at this point, I'm going to ask that you get pen and paper, Google, laptops, any implement that you need to use to memorize that the first reaction, glucose to glucose 6-phosphate, is a phosphorylation reaction, and it's done by hexokinase. We're going to discuss this a little bit more in the next slides. This is a energetically unfavorable reaction and is made favorable by the consumption of one ATP molecule. Therefore, the phosphorylation of glucose is coupled with the hydrolysis of ATP, and here it is. The first reaction is an example of something that you've talked about quite extensively in the first part of the class, of how enzymes have evolved these beautiful structures that can carry out coupled reactions such that an energetically unfavorable reaction, one that increases the internal energy of glucose to the high or internal energy of glucose 6-phosphate by hydrolyzing the ATP. The next one is a simple isomerization or rearrangement reaction. This is a spontaneous reaction. It doesn't happen with much change in energy. It's controlled by mass action. It's catalyzed by phosphoglucoisomerase. Reaction number 3, fructose 6-phosphate, is again phosphorylated to produce 1,6-bisphosphate. This is again done by a kinase. This is a transferase, if you remember. That would be the class 1 of the enzymes, of the 6 enzyme classes that we discussed last time. And it's specifically called phosphofructokinase. It's a major control point in glycolysis. It's not only that it catalyzes or couples the unfavorable phosphorylation of fructose 6-phosphate, but it's also the rate limiting step, the slowest step in the entire pathway. Reaction number 4 is actually the cleavage of the fructose bisphosphate, from which glycolysis takes its name, glycolysis. This is the lysis. Aldolase is a lyase, and I'm going to invite you to look up what class of enzyme that is. And then dihydroxyacetone phosphate can isomerize freely and spontaneously into glycerol 3-phosphate. These exist in equilibrium, and if you were in a test tube, probably the reaction would stop here. However, glycerol 3-phosphate is very much used up by the high concentration of NAD. NAD to NADH in a cell is anywhere from 700-800 per 1, 7-8-fold more. So this high concentration of NAD, or high ratio rather, makes this oxidation and phosphorylation into bisphosphoglycerate. So the continuous consumption of glycerol 3-phosphate is going to continuously pull mass, continually consume glucose, so that the next reaction is because this is being consumed by the next reaction. All right. So with bisphosphoglycerate, now this is a very high energy phosphate, meaning its energy of hydrolysis can not only phosphorylate ADP into ATP, but also release some energy such that the reaction occurs spontaneously in this direction. The next reaction, number 8, is an isomerization reaction by phosphoglycerate mutase. The phosphate from position 3 is being moved to position 2. And then a dehydration by enolase, which is represented here, increases the reactivity of PEP bonds. PEP again is like BPG, a high energy phosphate, meaning it can serve to phosphorylate ADP into one molecule of ATP by substrate level phosphorylation by pyruvate kinase, which again couples this reaction to make pyruvate and also exudes a substantial amount of energy, such that this, again, step 10 of glycolysis is irreversible. And this is how it just cannot, this pathway cannot ever backflow. And actually any other pathway cannot backflow. All pathways are spontaneous in one direction. And in the case of glycolysis, glucose will always form pyruvate through glycolysis. So some people will ask, well, can cell make glucose from pyruvate, meaning going the other way? And the answer is yes, but not with this glycolytic pathway. It's going to have to use a different pathway to get there, right? So it's not going to be a reversible reaction or reversible pathway. There is no such thing. All pathways are unidirectional spontaneously as written. And I'm going to say this so many times that you're going to be able to teach this to other people, I hope. All right. So I promised to talk a little bit about, how are we doing on time? We're doing good. I promised to talk a little bit about conservation of energy, and not only conservation, but also the directionality of the various reactions in this pathway. So here on the right-hand side, you really have the exact same pathway, molecules, intermediates, metabolites, enzymes, as in the previous one. I like this in particular because it also gives you a more explicit way that basically from one glucose molecule, because of the aldolase reaction, we obtain two C3 compounds, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Then from now on, we're going to have two molecules of each of the intermediates per glucose. So if you are to write the mass conservation, it's going to be one glucose, which is C6, carbon 6, into 2 pyruvate, which is 2 times 3 carbon pyruvates, 6 carbons. The carbons add up. And here's the oxygens, where oxygens happen. Here's the phosphate. And of course, ADP and ATP that we're going to keep track very much of. I am delighted that Dr. Kosman actually discussed with you the difference in examples, the difference between delta G0 prime, which is the standard free energy for each of these reactions, the 10 reactions of glycolysis with the 10 enzymes that catalyze each of those reactions. And if you were to do 1 mole of glucose with 1 mole of glucose 6-phosphate at pH 7 at the temperature of standard temperature, then this reaction will be very, very irreversible. I shouldn't say very irreversible. It's going to occur with a large thermal component. It's going to be spontaneous, it's going to be irreversible, it's going to release a lot of free energy. You're going to see different numbers reported as delta G0 for these compounds, and they're just going to differ probably by the conditions in which these were. It's not a big difference. Let me just say that. However, remember delta G takes into consideration the actual concentration of glucose and glucose 6-phosphate and also of ATP and ADP in the cell. Well, ATP is somewhere 5 millimolar, glucose is somewhere 5 millimolar. So it's not the molar concentration in which the standard conditions have been measured. If you compare the delta G0 prime with the delta G, the most important and striking feature is that the delta G is always negative. What does that mean? It means that even if, for example, the reaction catalyzed by aldolase doesn't really want to happen, its equilibrium is going to be very far to the products. The concentration of the products in the cell being very low are going to drive the reaction by max action to happen in the direction as written, with not a large excess of free energy, but sufficient such so that it keeps going, right? And then if you add it all up, you're going to see that the entire pathway releases quite a bit of energy, and therefore it's spontaneous. So this is represented here with the release in energy at each step, and you're seeing that basically at every single step, energy is either released or maintained into a sort of equilibrium here. Several points that I want to make here is the one that I already made, and I will repeat at all upcoming pathways, is that in vivo, all reactions are spontaneous, exergonic, right? They are made somehow to happen by either max action or by investing energy, by coupling them, tricks of the trade. In glycolysis, 1, 3, and 10 are irreversible, highly exergonic, right? And I highlighted these in red here. 16, minus 16, minus 14, minus 30. That's half an ATP. That's a full ATP just released, okay? And 3 is the rate limiting step, either the slowest or and it's also the commitment step. And I'm going to talk about that in a little bit because that's important. The last point that I want to make on this slide is to comment that in this series, in this linear series of reactions, dihydroxyacetone phosphate and glycerate 3-phosphate are in very low concentrations. They are in 1,000-fold less concentration than all the main characters here. Glucose is 5 millimolar, ATP is 5 millimolar, NAD is millimolar. These intermediates here are micromolar concentrations. So you can imagine that any time that the glyceraldehyde 3-phosphate is being used, where is it coming from? It's going to have to isomerase the dihydroxyacetone phosphate. And if this concentration is going really low, then it moves by mass action, this aldolase reaction, to break down more of the fructose 1,6-phosphate to make more dihydroxyacetone phosphate. So on and so forth. Although the internal concentrations are somewhat constant in the micromolar range, there is a homeostasis of this energy. They are not always the same molecule. More and different glucose is being broken down, lysed at all times. So for example, if you were to use a carbon labeled glucose, you're going to see it going through and appear that we would see that labeled carbon into pyruvate fairly rapidly. Although the concentration of dihydroxyacetone phosphate and glycerol, this high triphosphate, didn't change that much, right? And so it's pretty much a like a I'll explain that some more at another time. The last thing that I want to show you here, so PEP hydrolysis energy is 62 kilojoule per moles. So this reaction here by phosphoenol PEP kinase, pyruvate kinase, I'm sorry, it's not only irreversible, it's not only making ATP, meaning 30 kilojoules are going to be stored, quote unquote, into ATP, but it also releases 30 kilojoules of 30 kilojoules of energy per mole to make the reaction irreversible. So this is going to always drive the reaction. Okay, so if I go back here, let me see if I can go back. So basically, I'm gonna ask that you totally understand this 1, 2, 3, 4, 5, 6, 7 slides, okay? Several points about the major control points of glycolysis, and guess what? They are the 3 irreversible steps catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase. A couple of concepts and important information about each of them. We already discussed that reaction is irreversible. We're not going to go into the why in some textbooks it's 16, in other textbooks it's 20, based on the assumptions that they're making. It's largely exothermic, therefore irreversible, and it is a major control point. The flux is sensitive to ATP and ADP ratio by energy charge, because remember, ATP is a substrate. However, ATP as a substrate is going to have less of an effect if the reaction is already very much shifted towards the product. What really controls this reaction is allosteric feedback inhibition by glucose 6-phosphate. And we're going to discuss an exception for that. Hexokinase exists as 4 isozymes, very originally called 1 through 4. They differ in sequence, in expression, in kinetics, and in substrate specificity. Hexokinase 1 and 3 have very low Km. And therefore, if their Km is 0.2 and glucose is 5 millimolar, you can calculate that the reaction is going to always be at saturation. And these 3 enzymes are sensitive to allosteric inhibition, and it's really a product inhibition or a feedback inhibition, because glucose 6-phosphate is the product of this reaction. An exception is hexokinase 4, also called glucokinase, which has a very low affinity, 10 millimolar, right? So basically at 5 millimolar glucose, that's somewhere around its EC50. It's a low affinity, high capacity. So if it's on its still linear part, any increase in glucose concentration will increase its capacity, right? So it's going to be very sensitive to glucose concentration. Is specifically expressed in liver and pancreas, and it's a major control point for blood glucose homeostasis, and we're going to talk about it in several lectures going forward. Phosphofructokinase, again, catalyzes an irreversible reaction and also the rate limiting step. This is it's the most important or the most influential control point for glycolysis, and that's because it's also the commitment step. What does it mean to be a commitment step? It's the point where the only way of catabolizing that substrate is through glycolysis. It cannot turn into anything else. Why isn't it the why isn't the hexokinase the commitment step? And that the answer is hexokinase, by phosphorylating glucose, it forces it to stay in the cell. Glucose 6-phosphate cannot use glucose transporters to get out of the cell. So hexokinase immobilizes or tags glucose for cellular consumption. It traps it in the cell. In contrast, phosphofructokinase commits glucose to glycolysis. Glucose 6-phosphate can do other things. It can go on and make pentoses into an anabolic reaction. It can go and make glycogen in an anabolic reaction. So glucose, at the step of glucose 6-phosphate, glucose is not yet committed to glycolysis, to being broken down into pyruvate. However, phosphofructokinase does that. Once glucose is metabolized to fructose 1,6-phosphate, these 6 carbons, their fate is sealed. It has to be catabolized through glycolysis. So this is why phosphofructokinase is called the commitment step, and also it's the rate limiting step because it's the slowest of the reaction. It's controlled by AMP and ATP. So basically it has 2 heterotropic binding sites, meaning aside from its site where ATP is a substrate, so aside from the catalytic site, it also binds ATP at a distant or allosteric site, and that offers allosteric feedback inhibition. We're going to talk about this some more. Citrate is also an allosteric inhibitor, and that's because it tells the cell that if the citrate concentration is high, it means that the cell has other fuels to make ATP in a more efficient way, and that is through the TCA cycle by catabolizing lipids. So PFK is allosterically activated by high levels of AMP and allosterically inhibited by high levels of ATP and citrate. Lastly, pyruvate kinase catalyzes the most exergonic of the glycolytic reaction. This is the last step. It drives the flux through the entire pathway. It's a substrate level phosphorylation at the expense of PEP hydrolysis, and ATP is a product, so it binds in the active site. However, it also has very complex allosteric control. Fructose 1,6-bisphosphate is a feed-forward activator, an allosteric site. AMP is an allosteric activator, and ATP, by binding at an allosteric site, is an inhibitor. So that would be feedback inhibition. And acetyl-CoA is also a feedback inhibitor as an alternative fuel. This may seem like a lot right now. In several lectures, we're going to come back and explain this more and more and add layers of understanding, so that hopefully by the end of the 12 lectures, this really is going to make a concert. And you're going to be able to explain how the various pathways work with each other and what is really the mechanism by which this control occurs. I have already mentioned that pyruvate is a key metabolite in the cellular economy. And key metabolites, or nodes, right? I used the word nodes earlier, that by definition participate in multiple metabolic reactions that are part of different pathways. Therefore, they connect several pathways, and they're situated at metabolic nodes. And I'm giving you some examples here that pyruvate can be a substrate in anaerobic respiration. It can be a substrate or a main fuel in aerobic respiration, a substrate in lipid and amino acid biosynthesis. And we are going to discuss these reactions and their enzymes, their locales, and regulations in the future. For now, we're going to focus specifically in the pathway, in the last several reactions that that finalize the anaerobic respiration or fermentative fate of glucose or pyruvate. So anaerobic respiration is also called fermentation, meaning a respiration without oxygen, a respiration at the expense of some fuel, and in case of many animals, glucose is that fuel. So fermentations are enzyme catalyzed metabolic pathways that use pyruvate as the terminal electron acceptor to achieve fast and sustainable ATP production from glucose oxidation in situation where oxygen is low. Like, for example, the hypoxic muscle. Some of you already studied cancer cell. If they're hypoxic, they are going to use fermentation or anaerobic glycolysis. Certain cells are obligate anaerobes, meaning they don't have mitochondria. So basically, they don't have the machinery that does the aerobic oxidation of pyruvate, and therefore they're obligate fermentative cells, and these are the red blood cells or erythrocytes. The chemical strategy is to regenerate the NAD consumed in glycolysis by the glyceraldehyde phosphate dehydrogenase at the expense of reducing pyruvic acid. And depending on the enzyme present, the final product can be lactic acid, acetic acid, or ethanol. These end products or waste products are excreted or eliminated in the medium. I want to stop here for a second and make the point that the 10 glycolytic enzymes that we just discussed glucose to pyruvate. Those are conserved and exist in all cells. We said ubiquitous, we said universal, we said conserved. Fermentations are not. Fermentations are going to occur if there is no oxygen and if there are expressed in this particular cell that we're talking about the enzymes that can oxidize pyruvate to regenerate NAD. Let's see how and when that happens. So one of the most important fermentations that in this course I would like to focus on is the lactic fermentation, because it happens a lot. Erythrocytes, red blood cells are, and I give you some other examples here, are obligate anaerobes, and they get their energy from lactic fermentation. Also, the skeletal muscle depends on lactic fermentation anytime when its energy demands exceed the amount of ATP that can be obtained through aerobic respiration. So by definition, lactic fermentation is a metabolic pathway that uses glucose anaerobically to produce ATP and lactate. Its purpose is to make ATP and regenerate NAD necessary to continue catabolizing glucose to lactate. It requires the enzyme lactate dehydrogenase, which is expressed in the cytosol of most all tissues. It catalyzes a reversible reaction. Okay? It is a central metabolic pathway in the hypoxic myocytes, and it's the only energy-generating pathway in erythrocytes. LAC is a waste product for that particular cell. It's going to be excreted by muscle and the red blood cells into the bloodstream in the serum, and it can be taken up by liver, where it can, let's say, be converted to pyruvate or made into amino acids. In liver, many things can happen to pyruvate. But in the cells that die and live by lactic fermentation, the end product of glucose fermentation is going to be lactate. And I'm giving here some examples of just how important this reaction is, and therefore lactate dehydrogenase. No wonder lactate dehydrogenase has been studied for many years, and actually in studying lactate dehydrogenase, scientists have found out for the first time the concept of isoenzyme, meaning polypeptide that differ just slightly in their amino acid sequence. They're obviously encoded by separate genes. And to give you the example here, lactate dehydrogenase is a tetrameric protein, meaning it's assembled by 4 protomers from polypeptides encoded by 2 genes, separate. Let's call one M, which is mostly in muscle, and one H, which is mostly in heart. These, in all other tissues other than muscle and heart, both of them are expressed in various levels and also across development. And you're going to see here that LDH5, which is M4, is the first one. M4 is the first to be expressed, and much later, and in the adult, we have the entire H4 or LDH1 expressed in heart. So developmentally and tissue regulation of the expression of M and H gene genes that encode M and H peptides that can get together as H4 tetramers, M4 tetramers, or a combination of H and M tetramers to make 5 distinct forms of enzymes. And this panel here shows you the development, the regulation during development, and this graph over here gives you the tissue-specific expression of the various isoforms. And in the next slide, it's actually, I'm showing you an experiment that takes advantage, leverages the fact that these 5 enzymes differ very little. Actually, H and M peptides differ by 2 amino acids. So this is an enzyme that has, 300 amino acids, and just 2 are different. They're not visibly different, right? They're very similar, yet we can, or scientists, others have found ways to separate them. For example, by size exclusion chromatography, which separates them by size. And here's the elution profile, such that LDH5 elutes first. It's the largest, and it's not going to be retained. And all the other ones are retained more and more and more. And you're going to see that actually LDH3 elutes as 3 separate peaks. So these sort of differ a little bit. Gel electrophoresis separates proteins by size and charge, right? For example, you take cytosol from retina and you load it onto a gel and you force these proteins to migrate through the gel towards the positive pole, the anode, and they're going to migrate according to their charge and also according to their filtration through the gel. Okay, and this is how preparations from retina have 1, 2, 3, 4 types of enzymes. They don't have LDH1. Muscle basically has only M4, and heart has a variety of them, and brain has a variety of them. So you basically do the experiment to see which combination this particular tissue has from this particular patient that has been treated with this particular drug. So it's a really very sophisticated and high resolution way to determine what kind of enzymes are expressed. It's also very good. I'm not sure I'm telling you, okay, I'm not telling you there, but it's very efficient way of diagnosis, for example, muscle breakdown or liver breakdown or heart breakdown, because the serum has a certain composition in terms of LDH profile, and clearly if you're starting to see muscle LDH in serum, immediately you're going to know that some muscle breakdown has happened, or heart attack. You can diagnose by a quickly profiling LDH in blood to see if any heart cells or brain cells have burst out and liberated their cytosolic LDH into the extracellular fluid, in this case, the serum. So moving on to alcoholic fermentation, this is a pathway that oxidizes glucose also anaerobically, but instead of producing lactate, it decarboxylates pyruvate. Okay, so this is a stage 3 pathway, right? Remember, stage 2 is going to be monomers to metabolites, as stage 3 will be metabolites or monomers to inorganic. So CO2 is already an inorganic molecule, right? So from C3 pyruvate, we get acetaldehyde. I'm sorry, from C3 pyruvate to C2 acetaldehyde. And then at the expense of acetaldehyde, NAD is being regenerated with the formation of ethanol. It's an important industrial process. It doesn't happen this way in mammalian cells. And one important thing that I should say is that the fact that CO2 is a gas makes this reaction fully irreversible, in that acetaldehyde cannot incorporate CO2 to make pyruvate. We're going to learn of a different enzyme and different mechanism to do that. However, ADH is a ubiquitous housekeeping enzyme. It's used in the opposite direction. So it's going to be used to dispose of ethanol. That's especially ethanol, as you all know, is a CNS depressant and toxin, and therefore ethanol, which can be either ingested or produced by various pathways, has to be detoxified. In humans, kidney and liver are the primary tissues for ethanol detoxification or catabolism, and it's also most prone to acetaldehyde damage, like for example in individuals who chronically consume large amounts of ethanol. So what are the enzymes involved in alcoholic fermentation? We talked about lactate dehydrogenase for lactic fermentation. Here we're going to need two enzymes. One is pyruvate decarboxylase, and I'm giving you a number of some information about it. Oh, it's a funny fact that the fact that CO2 is being released, this is how initially these reactions were called fermentations. This is how enzymes were discovered as being ferments from yeast in the late 1800s. So we are still stuck with these names. So pyruvate decarboxylase, and we're going to talk some more about it. And alcohol dehydrogenase, which is ubiquitous in all cell types, but it works specifically in liver and in kidney. So, as I mentioned, in human tissue, the fact that we don't have pyruvate decarboxylase, human cells cannot produce ethanol from pyruvate. However, ethanol occurs from other pathways, and therefore it needs to be detoxified, and that's what alcohol dehydrogenase does. All right. And the last fermentation that I'm going to be talking about today is the acidic fermentation. It's also an anaerobic glycolytic pathway, occurs in certain bacteria. And in addition to the 10 glycolytic enzymes that are universal, ubiquitous, conserved, these organisms also express pyruvate decarboxylase, which decarboxylate pyruvate to acetaldehyde, and make acetaldehyde to ethanol, but also aldehyde to acetic acid. Okay, and this is the pathway where, for example, in industrial processes, sugar is going to be made into ethanol with yeast extracts by alcohol fermentation. And then bacteria are being added to make vinegar or extracts of bacteria to make acidic fermentation. All right. You're going to notice that for some enzymes, I give you the enzyme class. And this is not for you to memorize it, but rather this first number here, for first significant number, it tells you the enzyme class. So basically, if it's EC1, it's going to be a transferase. Obviously, all dehydrogenases are transferases, and we're going to see other examples especially in the quiz. I hope I bring this up. So, bazillion applications. Please give this a read. You probably know your own. With the pandemic, a lot of people started making beer and wine and baking. So certainly, many of you have firsthand benefit of the application of fermentations. Not to mention that every time you go to the gym and you strain your muscles to exert themselves more than aerobic respiration allows, you're going to produce lactic acid. So this is all interesting and certainly very important. A review of everything that we talked about. If at any time you find either in the objectives or in the review that there's something that perhaps I didn't address sufficiently or you're confused about, please bring it up on Friday during our live sessions in Zoom. And this is a cheat sheet for you that for any metabolic pathway to know its major life-sustaining purpose or its mission statements, describe the chemical strategy, the sequence of chemical reactions, name the enzymes, know the type of enzymes, that first number, the type of enzyme, it is a transferase, an isomerase, a ligase, a lyase. Describe the locale of where the enzymes are. Like, for example, for glycolysis, they're all cytosolic. They can work in the absence of mitochondria. Balance the energy, carbon, and electron content for each reaction. Explain reversibility, meaning how is the energy conservation and the spontaneity work. Enumerate the main regulatory mechanism, and in the cases that we get a chance to discuss the importance in facilitating scientific progress, like for example in the case of isoforms, clinical practice, like for example diagnostic of heart attacks with LDH enzymes in serum, or biotech application as in the production of vinegar, and we're hopefully going to discuss these for other pathways as well. For now, I know it's a lot and it comes fast at you. So we're going to discuss on Friday any questions that you have about lectures 1 and 2. And hopefully you'll post at any time questions on discussion board. I will start monitoring them right after your exam. Also, be sure to take the quiz before Thursday at 5. So based on your preliminary scores, I can prepare to emphasize or clarify any points that seem to have been confused or misunderstood, and I just want you to leave with a good understanding of metabolism. So I look forward to seeing you on Friday, and good luck with your quiz.